So I have so the Japanese case.
Anything that stands out to
Oh.
you, anything that you have questions about, right?
I think
I met someone who had taken this class before. So I need to talk to him because he said that he would help with like knowing what the professor actually wants.
so we have
For this week. But in the meantime,
but he's right now. Yeah. Why he's talking about this trip, but
all right, yeah, you're like, how are you? Traffic is like,
so they're all going. I'm just dropping them off. But then they're in different places. Yeah, but that's like 7 hours. 7 hours?
Yeah, so there's a lot so
We were both trying to convince
what it's like ultimately just imaging difference like eye imaging
Good morning, everyone. We will suspend the breakfast because if we at least don't say good morning, that's the only thing you have to say. Good morning. Good morning. Now we have energy. So it's my pleasure today to introduce Dr. Abdul Muneer. He's visiting us and participating in this Wallace Coulter seminar series. He's currently associate professor at the JFK University Medical Center, the Hackensack Meridian Health, the JFK University Medical Center, at the Department of JFK Neuroscience at the Institute. He got a Master in Science from Cochin University of Science and Technology in India and a PhD in molecular biology for the same university. And his expertise have been developing over the years around traumatic brain injury and developing intervention, understanding the mechanistic foundation of this disorder, and also developing intervention. So today we will enjoy the most advanced progress in this field from Dr. Muneer. So let's welcome Dr. Muneer.
Thank you, Doctor, for your nice introduction. Good morning, all.
Good morning.
Very nice to meet you all here, and welcome for my presentation. So today I'm going to talk about the topic, transmigration of leukocytes and the formation of neutrophil extracellular traps in TBI, a novel therapeutic target. So before coming to the mechanisms of transmigration of leukocytes and formation of neutrophil extracellular traps, I'm going to give a brief introduction about TBI. What is TBI? Traumatic brain is a complex injury with a broad spectrum of symptoms and disabilities. It changes personality and mental abilities of affected individuals. It impairs cognitive functions, leads to permanent disabilities, coma stage, sometimes sudden death. Approximately 2.1 million people's lives are affected by TBI every year in the US. What are the leading causes of TBI? Falls comes number one, 35.2% of TBIs are because of falls. Motor vehicle traffic crashes, 27.3%. Sports concussion, 16.5%. Assault, 11%. And other unknown, including blast injury, 10%. Who is at highest risk for TBI? The age group at highest risk for TBI is 0 to 4 years old, 15 to 19 years old, and adults age 65 years and older. The age group 0 to 4 years old and age 65 years and older is mainly because of falls. The teenagers 15 to 19 years old group is mainly because of motor vehicle traffic crashes and sports concussion. 78% of TB are affected by males and only 22% are females. This slide shows the symptoms of brain injury with respect to the injury of different parts of the brain. If injury in parietal lobe, it affects motor sensory, pain, touch, sensation, spatial orientation. If injury is in frontal lobe, it affects judgment, planning, concentration. If injury in occipital lobe, it mainly affects vision. If injury in temporal lobe, it affects memory, speech, auditory, and visual perception. If it is in cerebellum, it affects coordination, balance, injury, and muscle tone. If injury in brainstem, it affects the regulation of vital functions, respiration, heart rate, and blood pressure. TBI is categorized as primary and secondary injuries. Primary injury is a result of mechanical forces applied to the skull and brain that leads to skull fracture, brain contusion, rupturing of blood vessels, and intracranial hemorrhage. Secondary injuries It's mainly characterized by complex cascades of biochemical events that leads to oxidative stress, blood-brain barrier disruption, neuroinflammation, neurodegeneration, ischemia, and so on. The main events of primary injury is mechanical tissue deformation resulting in necrotic cell death, shearing and tearing of blood vessels, neurons, glia, and axons, and this leads to secondary injury. The main events of secondary injury is excitatory amino acid release, calcium influx, induction of free radicals, cytokines and chemokines activation, changes in several gene expressions, mitochondrial damage. These all events leads to blood-brain barrier damage, edema, increased intracranial pressure, altered cerebral blood flow, ischemia, and hypoxia, and increase in lactate energy deficit. And this finally leads to cell death and functional deficit, like it affects, it impairs the cognitive function, sensory motor function, etc. TB models. We use in vitro and in vivo models in my lab. So in vitro model is called a structure injury model. The device we use for structure injury is called the cell injury control 2 device. So we use a specialized plate for culturing cells is called a silicone membrane plate. This is not normal 6-well plate, this is silicone membrane plate. The bottom is a soft silicone membrane. So we culture cells on top of this plate and pass gas through the cells. When gas passes, the membrane will bulge like a balloon and all cells get stretched. That's called a stretch injury. And in vivo the model is called the fluid percussion injury model. So this can see the glass tube is filled with fluid. We usually fill with buffer, and when this pendulum hit to this glass tube, the fluid creates the pressure, and this pressure connects with the brain of animals and creates injury. So we can also adjust this injury with a different, mild, moderate, and severe, according to where we hit this pendulum. Also, this is computerized model. Similarly, we can also inflict mild, moderate, and severe injury in vitro models too. So this is the injury site. This is schematical representation of injury site. We usually conduct injury in the parietal lobe. This blue color is the frontal lobe, and green is the hippocampus. The red is injury site. So this is stereotactic injection frame. Now coming to transmigration of leukocytes to the brain. So when we talk about transmigration of leukocytes to the brain, the main reason for this transmigration is dysfunctioning of blood-brain barrier. So what is blood-brain barrier? It is a form of an array of brain endothelial cells. It is a highly selective barrier, allows ions, water, some gases, glucose, and amino acids, and it prevents lipophilic molecules, toxins, microorganisms. This is cross-section of brain microvessels. So the layer of endothelial cells surrounds with astrocytes and feeds and pericytes. Astrocytes and pericytes give structural stability to this blood-brain barrier. This is a cross-section of blood-brain barrier. This is all called neurovascular unit. So, when coming to the tight junction proteins, the two adjacent endothelial cells are stitched together with a certain protein is called a tight junction proteins. There are different types of tight junction proteins. A smaller transmembrane protein is like occludin, claudin, and ZO1. This connects these two endothelial cells together. And there are another type of protein is called heterin junctions. Cadherin, beta-catenin, alpha-catenin. These are all examples of heterin junction proteins. So now coming to the role of pericyte in BBB stability. We recently worked and published one paper in experimental neurology. Impairment of pericyte endothelium crosstalk lead to blood-brain barrier dysfunction following traumatic brain injury. It's a highly cited paper. So in pericyte has a significant role in neurovascular unit and giving the stability and integrity of the blood-brain barrier. So, and it also has the function in the formation of tight junction proteins of the BBB. So in brain injury, the blood-brain barrier affected, so it compromised blood-brain barrier, and the cells surrounding the cells of the endothelial cells get loosened, and here we do the immunostaining of this neurovascular unit after 7 and 15 psi injury. So here you can see the cells are loosely arranged, they become very loosely arranged in the brain injury samples. So GFF is a marker of astrocytes, and the PDGFR beta is a marker of pericyte. PDGFR beta is platelet-derived growth factor beta. So this is schematic representation how endothelial cells and pericyte connect together and gives structural stability to the blood-brain barrier. So here in between pericyte and endothelial cells, you can see this basement membrane in between. This basement membrane, in basement membrane, you can see that there are certain integrins tightly joined endothelial cells and pericyte. And also there are different types of proteins called NK3, connection 43. This brings together endothelial cells and pericyte. This is a Western blotting expression of PDGFR beta. This PDGFR beta expressed in pericyte. So in control, this highly expressed, and injury after 12 hours, 24 hours, and 48 hours. You can see this expression level is significantly reduced. And another aspect, PDGFB is a ligand of PDGFR beta, which is present in endothelial cells. And its receptor, PDGFR beta, is expressed in pericyte. And this signaling is necessary for pericytal recruitment during angiogenesis. Now coming to blood-brain barrier leakage. So during injury, there's a chance of compromising blood-brain barrier, and there's a chance of leakage, the blood cells to the brain part. So there are many reasons for the leakage of blood cells to the brain part. It's mainly oxidative stress, MMP activations, neuroinflammation, and tight junction protein degradation, and so on. So this right side, you can see the immunostaining of CD68 is a macrophage marker in mild fluid percussion injury after 6 hours and 24 hours. So here, CD68 expressed abdominal side of, that means the brain side of, brain part of the microvessel, expressed very highly in fluid percussion injury after 6 and 24 hours. And we call localized with 1 millibrand factor is a specific marker for microvessel. So analysis of blood-brain barrier permeability, we use different methods to analyze blood-brain barrier permeability. First, we use 2 different dyes called sodium fluorescein and Evans blue, 2 different. Sodium fluorescein is a smaller molecular weight molecule, and Evans blue is a higher molecule, around 900 kilodalton. Sodium fluorescein is 300 kilodalton. And we infuse the mixture of these 2 dyes through the exposed carotid artery, and we analyze its present in brain samples. So here in this graph you can see, like in TBA sample, the presence of sodium fluorescein and Evans blue significantly high. And we also use another method by ELISA of S100 beta and NSE, neuronal specific enolase. These 2 markers will be very high in TBA samples. Here, for validating, we use another method to show the transmigration of blood cells in the brain part. What we use, we collect bone marrow and culture monocytes, so that monocytes go into the macrophage. And this we label with the fluor3 is a fluorescent labeled dye, and labeled with, and this fluor3 labeled macrophage infused to the carotid artery, and we analyze its presence in the brain part. And for validating another method, we use GFE mouse macrophage, which is a green fluorescent cells. This macrophage we infuse through the carotid artery and we analyze under microscope, under fluorescent microscope, or we also stain with the anti-GFE antibody for confirmation. So cerebral blood flow, we also analyze cerebral blood flow. The device is called a laser Doppler system. So usually in TBI, the blood flow is very highly diminished, actually, significantly diminished. In normal cases, it's around in between 150 and 300. And in injury cases, it comes like close to 75. So now comes to the role of addition molecules in transmigration of blood cells. There are different addition molecules like ICAM, VCAM, P-selectin, E-selectin, but each addition molecule has its own role in transmigration of blood cells. There are 3 steps of transmigration. The first one is rolling of blood cells. In this one, P-selectin and P-selectin glycoprotein 1 involved mainly. The second one is activation of foam addition. In this one, the blood cells adhere with the wall of the brain microvessels with the help of ICAM1 and VCAM and its receptor. And third one is transmigration of diabetes is final. So VCAM is involved in the transmigration of blood cells to the brain part. So this is a schematic representation of this. The role of addition molecules in the transmigration of blood cells. Now, we laboratory study, we extensively studied about the role of ICAM1 in transmigration of leukocytes to the brain in TBI. ICAM is an intracellular addition molecule, is identified as an initiator of neuroinflammatory responses that leads to neurodegeneration and cognitive sensory motor deficit in several pathophysiological conditions. ICAM gene has a significant role in the regulation of vascular permeability, impairment of blood-brain barrier, and transmigration of leukocytes to the brain. So ICAM1 present in endothelial cells and its receptors, mainly 2 receptors, the LFA1, leukocyte function associated antigen, and MAC1. These 2 receptors present in leukocytes, especially neutrophils, monocytes, and macrophages. This is the immunostaining of ICAM1 and its receptor LFA1 in control and injured samples. And also we validated this expression in ICAM-KO animals, knockout animals. So expression of ICAM1 is very high in injured samples. Similarly, expression of LFA1 also very high in injured samples compared to uninjured animals. This we recently published in the eNeuro in 2021. So for validating the role of ICAM1 in gene deletion, in transmigration of leukocytes, we delete this ICAM1 by using CRISPR-Cas9 tool. So this is a vector for ICAM1 CRISPR-Cas9. We use the AAV vector, serotype 9, and then we analyze the expression level of ICAM1 in vitro and in vivo. In the first graph, you can see the expression level of ICAM1 is very high in injured samples. When we delete by using ICAM CRISPR, the expression level of this ICAM significantly reduced. Similarly, in vivo, the same trend of result we observe. This is mRNA expression of ICAM. So in injured, so ICAM expression is significantly high. And when we delete ICAM using ICAM CRISPR, is significantly reduced. This we published in the Journal of Neuroscience very recently. So this is for validating what we did. We separate, as I told you just before, we separated GFE monocytes from GFE, like transgenic animals, and infuse these GFE cells to the carotid artery of control and injured samples. And also we compare with the ICAM knockout. So here, the large number of GFE positive cells we observed in injured sample, and we also localized with the MAC1. MAC1 is a blood cell marker. So it is a neutrophil marker. So you can see here, GFE monocyte positive cells is very high in injured samples, and when we treat with ICAM CRISPR, it's significantly reduced. And in KO animals, we cannot see much GFE positive cells. So this is cover image, Journal of Neuroscience published from our work in March issue, and also this paper they highlighted as a highlight of research in that particular issue. So coming to my NNDS R1 project, this started on March 2024. So this all transmigration research, ICAM, the role of ICAM on transmigration of leukocytes, finally leads to this R1 project. This is the continuation of the transmigration work. So here, the project title is called Neutrophil Extracellular Traps and Associated Pathogenesis in TBI, a novel peptide therapeutic strategy. The goal of this project is to develop a therapeutic strategy for TBI, targeting net formation by blocking the biological activity of PAD4 using an antagonistic peptide. PAD4 is an enzyme is called peptidyl arginine deaminase. The hypothesis of this project is that inhibition of PAD4 using PAD4 antagonistic peptide, briefly called PAD4 antagonistic peptide, will suppress the formation of NET and NET-induced thrombosis and promote neovascularization and functional recovery after TBI. There are three specific aims in this project. Specific aim 1 is to study the therapeutic regulation of formation of neutrophil extracellular traps using PAD4 antagonistic peptide. So mainly the mechanisms of how a NET forms after TBI. And also we use PAD4 antagonistic peptide for blocking this formation of NET. Specific aim 2 to study the mechanisms of NET-induced thrombosis and therapeutic role of PAD4 antagonistic peptide in depleting NET-dependent thrombosis in TBI. And specific aim 3 is to investigate the therapeutic role of PAD4 antagonistic peptide in promoting functional recovery in TBI. This means mainly behavioral studies. So coming to this project, specific aim 1. So first of all, what is neutrophil extracellular traps? Activation of neutrophil leukocytes, especially neutrophil, causes the release of nuclear and granular contents to form extensive web-like structure of DNA called neutrophil extracellular trap. These extracellular strands of decondensed DNA in complex with histones and neutrophil granule proteins, including neutrophil elastase, cathepsin G, and MPU. These are the main markers of neutrophil extracellular traps. Neutrophil elastase, cathepsin G, MPU. Another important marker is the H3 citrulline. So these nets have been associated with autoimmune disorders, cardiovascular, pulmonary diseases, inflammation, thrombosis, and even cancer. So the formation of NET is called netosis. There are two mechanisms of formation of NET. The first one is the leukocytes or neutrophils. The nuclear delocalization happens and disassembly of nuclear envelopes occurs. Then cellular depolarization and chromatin decondensation, then plasma membrane rupture and release nets. In another step, another mechanisms, non-lytic netosis, called non-lytic netosis. In this one, the neutrophil itself, granulation and expulsion of nuclear chromatin, everything happens. Then extracellularly, these all nets assemble, and finally this becomes like a engulfed in phagocytotic cytoplasm, and this ingest into the lysosomes and other cells. Peptidyl arginine deaminase. So this is a crucial factor of formation of neutrophil extracellular trap. This peptidyl arginine deaminase is an enzyme that plays an important role in gene expression, turning out genetic coding to functional products in the body. So here is the enzyme is called PAD4. PAD4 is like the main converts, like the main reason for the formation of the net. So here the mechanism is PAD4 convert peptidyl arginine into peptidyl citrulline. Histone modifying enzyme that is crucial for net formation. It converts arginine to citrulline and promote deamination of arginine residues in histones and may regulate transcription in the context of chromatin. This is immunostaining of PAD4 in neurons. PAD4 expressed in different types of cells, including neurons, endothelial cells, and astrocytes. We didn't see much in microglia. And we call our class PAD4 with UN is specific marker for neurons. Peptide therapy. Peptides are short linear chains of amino acid, a unique class of pharmaceutical compounds molecularly points between small molecules and proteins, biochemically and therapeutically distinct from both. So advantages peptide therapy is high potency of action, high target specificity and selectivity, wide range of target, low accumulation of tissues, high biological and chemical diversity, and low side effects. So we designed different 4 peptides against the PAD4. It's called PAP1, PAP2, PAP3, and PAD4. And compared the result with the control peptide. It's a random sequence. So PAP2 in our preliminary experiment, PAP2 shows like very good results. So we select PAP2. And this peptide we joined with the TAT peptide is derived from the transactivator of transcription of HIV and is a cell-penetrating peptide. For penetrating to the cells, we join this TAT sequence with this PAD4 peptide. So the result shows we injected this PAD, PAP means PAD4 antagonistic peptide subcutaneously and analyze the result. Here, this is the immunostaining of net marker H3 citrulline and blood cell marker LY60. You can see like in injured samples, H3 citrulline is highly expressed. This is a large portion of this one. And you can see in PAD4 KO, you can see some neutrophils, but there is no H3 citrulline. That means the formation of net is totally absent in PAD4 KO animals. So this is percentage of LY60 positive cells and percentage of H3 citrulline positive cells. In injured samples, H3 citrulline is significantly high, and when we treated with the PAP, it's significantly reduced. And number of H3 positive cells is very less in PAD4 knockout animal samples. Now coming to specific aim 2, net causes thrombosis. So this is the first study in TBI, how TBI-induced net leads to thrombosis. So there are different mechanisms here. We are studying like there's one step for forming the thrombosis is the activated neutrophils joined with the nets and platelets and aggregates and attach to the wall of the microvessel. So the enzyme thrombin is helping in this aggregation and formation of thrombosis. So this is how neutrophil, platelet, and endothelial cells, the 3 different types of cells, aggregates together during thrombosis. So here first, platelet and endothelial cells joined with the platelet receptor GP1 beta and GP2 beta 3A with the 1 milligram factor of endothelial cells. And similarly, platelet and neutrophils join together with the different ligands and receptors, mainly PSGL1 with P selective and GP1 beta with MAC1 and GP2 beta 3 and SLC44A2 and CD40 bit to CD40 ligand and ICAM2 bit its receptor LFA1. Similarly, neutrophil and endothelial cells join with ICAM and its receptor LFA1 and MAC1 and BCAM with its receptor BLA4. So this is the how neutrophil, platelets and endothelial cells interact together and forms thrombosis. So here we analyze the role of PAD4 antagonistic peptide in depleting net-dependent thrombosis. In our result shows that there is a thromboxane mRNA. Thromboxane is an enzyme which is produced by platelets and this induces thrombosis. So thrombosine mRNA in injured sample is significantly high. When we treat it with the PAP, it is significantly reduced. And in KO, PAD4 KO is significantly less the expression of thromboxane mRNA. Similarly, this immunostaining of H3 citrulline is a net marker, and GP1 beta alpha is a platelet marker, and this shows the thrombosis in TBA. So here, H3 citrulline and GP1 beta alpha exactly co-localize in injured sample. You can see there are several significantly high number of clots in injured samples. And when we treated with the PAP, this number of clots significantly reduced. And very, very less number of clots we found in PAD4 KO animals. Functional recovery analysis. So after in the third specific case, what we did, after treating with the PAD4 antagonistic peptide, we analyzed sensory motor function by rotor rod, grid walk, balance beam test, and we analyzed spatial memory and learning by using Morris water maze and novel object recognition test, and we also analyzed anxiety-like behavior and depression-like behavior by light dark and open field test and suppressed preference test respectively. And this is the data of a rotor rod test. Here, the latency to fall, the time to fall from the rotor rod is called latency to fall, is very, very less in injured samples. When we treated with the PAP, is significantly increasing. Similarly, this is a light road dark test result. So here, the percentage of the time spent in light chamber is significantly less in injured samples. When we treated with the PAP, is significantly high. This is Morris water maze result, and we recently published in Journal of Neuroscience in 2024. So future direction of a NET project. So we plan to study the further progress of this NET project, mainly in the role of NET in atherosclerosis, how the lesion forms and plaque development and formation of atherosclerosis, and the role in pharmacology and any therapeutic strategy. That's all our plan. And in other aspects, we study the role of leukocyte-derived extracellular vesicle in TBA-induced BBB damage, neuroinflammation, and neurodegeneration, and we also plan to develop a possible therapeutic strategy. So here the main role of is two NET marker, MPO and cathepsin, and together with other protein like S100 beta and cytokine, this cause BBB damage and increase permeability. MPO binds to glycocalyx heparan sulfate, which induces the release of neutrophil granular proteinases to cause syndican-1 shedding and glycocalyx impairment. And cathepsin, another marker of a NET, is known to increase endothelial permeability to albumin through the detachment of plasminogen activator inhibitor 1. In addition, cathepsin G degrade weak heparin and impair blood-brain barrier. So other grand plan, we recently submitted the project number 1, role of piRNA in the pathogens of TBI. This is a very novel first study in TBI. So what is piRNA? Pi-V interacting RNA. PiRNA constitute one of the major class of small non-coding RNAs. Unlike microRNAs, a small interfering RNAs, piRNAs are longer, 21 to 35 nucleotides, and do not require dicer 1 or 2 for their production. So here in this project, our aim is to identify differentially expressed piRNAs and study their role in synaptic plasticity and in developing Alzheimer's disease character after mild TBI. So this project has been discussed in the NIH study sections and have to resubmit I'm planning to resubmit in February 2025. In our preliminary study, we identified a novel 1,849 piRNAs in mild TBI, and out of these, 16 piRNAs are significantly expressed. Among these 16 piRNA, 6 were upregulated and 10 were downregulated. We designed primers for the 6 upregulated piRNA. These are primers. You can see the bottom figure is primers of the 6 upregulated piRNAs. This first figure is an expression of PIV 1, 2, and 4. This PIV protein linked with piRNA in control sample and injury. So you can see in injury sample significantly high. And also we analyzed neurodegenerative marker annexin 4 and neurofilament light chain after blocking PIV 1, 2, and 4. You can see annexin 5 is significantly high in injured sample, but by blocking with the PIV2 significantly reduced. That means it has link with the neurodegeneration. Similarly, neurofilament light chain in injured sample is expression of neurofilament light chain is very, very less. When we block with PIV2 or PIV4 is significantly high. Neurofilament light chain is also considered as Alzheimer's markers. In cells or tissue samples, you can see this expression of neurofilament light chain is very less because it degrades. But this degraded neurofilament goes to the brain circulation, blood circulation, and you can see the expression or level of neurofilament light chain is very high in blood serum or plasma. So role of piRNA in the pathogens of TBIs, and here we also designed developed therapeutic strategy by designing antagonistic oligos. It's called antago-pill, similar to antago-mill for microRNA. So here piRNA we call it as antago-pill. So we designed to block piRNAs as certain antago-pills. This is 2 antago-pills for blocking piRNA 57522056 and another one in 4945081. And we analyzed electrical activity to determine the changes in the baseline synaptic transmission and memory-related long-term facilitation in the neuronal culture in the presence or absence of FIU. This is electrophysiology work. And second, and also we analyzed synaptic plasticity markers, CRUB2, DNA methyl transferase 3A, and CAMK2 beta, home scaffold protein 1, and BDNF, and also we analyze Alzheimer's markers. And another project, our plan is to study the mechanisms of developing Alzheimer's disease-like characters in mild TBI, a role of synaptogary and therapeutic strategy. This is first study in TBI, the role of synaptogary in Alzheimer's disease-like characters development. Publication plan. First one is an ARF2 activator peptide protects the brain from cerebral vascular dysfunction in alcohol ingestion. I do alcohol with around 30% alcohol research, and we are also planning to do synergistic effect of TB and alcohol. We already published one paper on that. And this, my current R21 grant work, this paper has been completed and it will be communicated in October next month. So NRF2 regulates leukocyte transmigration and neutrophil extracellular trap formation after traumatic brain injury. This paper of around 80% has been completed and will be communicated in January 2025. And paper number 3 and 4 has pending work at ICAM antagonistic peptide attenuate transmigration of leukocyte to the brain after traumatic brain. Similar to PAD4 antagonistic peptide, we also analyzed the role of ICAM1 antagonistic peptide. And this paper is under production. And fourth one is traumatic brain injury, neutrophil extracellular trap formation, novel therapeutic strategy, the current R1 project work. Conclusion, TBI is a major cause of death and disability in the United States. Transmigrational leukocytes is a major hallmark of neuroinflammation in TBI. Additional molecules have major roles in the transmigration of leukocytes to the brain. ICAM1 deletion attenuates the transmigration of leukocytes to the brain. Transmigration and activation of neutrophil causes the formation of NET. PAD4 enzyme has a crucial role in the formation of NET. We designed PAD4 antagonistic peptide, it's called PAP, to block the action of PAD4. PAP treatment reduces the number of NET marker positive cells. NET causes thrombosis, and PAP treatment reduces the thrombus formation in TBI. PAP treatment promotes sensory, motor, and cognitive functions after TBI. PAP treatment reduces TBI-induced psychological stress. So, Dr. Saikia, Preeta Rani, all postdoctoral fellows, Daniel Caruso, research technologist, Preeta Rani, recently she left the lab, Christina, Yemin, Ramdan, all undergraduate students. Funding, NIH, NDS R01 and R21, and New Jersey Brain Injury Commission. Thank you. Thank you very much.
Thank you very much, Dr. Muneer. We open the floor for questions.
So, the grant ones that you mentioned about for memory and cognitive, memory and cognitive assessment, you mentioned about novel object recognition test.
Yes.
Have you done that test?
Yes. We did, and we published in Journal of Neurology.
Days after the injury when you perform the test.
That's a good question. Behavioral studies, sometimes we cannot do immediately after injury because animals, it won't do much for the behavioral studies. But we plant a second day, 7th day, 14th day, till 1 month actually, every week. We studied, second day we avoided because of the animal was not doing very good after injury. So 7th day, 14th, 21st, and 28th day we did. We published, we can see
throughout the time there was a cognitive decline in the memory. I mean, there is decline in the memory.
It varies actually, that depends on number of days actually.
Question.
Good question, thank you.
You said that they were minimal side effects when you were studying and you were doing the test. Can you tell us what the side effects are?
Peptide, right?
Mm-hmm.
Peptide therapy?
Yes.
Yeah, we studied toxicity, the cell death in animals, and also in vitro we studied actually. The toxicity, cell death, and there is one method we use, I forgot. And also we studied neurodegenerative markers and toxicity to the animals and toxic to the cells, both we studied. Yeah. We published this in, this you are asking about PAD4, right? Yeah.
Any other question?
Good question.
Question?
So you mentioned in your introduction that parasites play a role in blood-brain barrier.
Yes.
So the morphology and coverage of parasites on the capillary network change dramatically. So we have encysting parasites at the beginning covering completely capillaries. These parasites covering almost none of the capillaries. So I was wondering if you have any take on this role of pericytes on blood-brain barrier as affected by atrophy capillary network. In other words, if the pericytes are important, why they're not covering completely the capillary of blood-brain function?
Okay, this here is what we found. Pericyte attached with the neurovascular unit and tightly it gives the integrity to the blood-brain barrier. But in TBA, what we found like the integrity loosens, but the attachment between pericyte and endothelial cells, the bond between these two types of cells is diminishes actually. The bond, it affects mainly bond. We published in 2019 this paper in experimental neurology. So, and some other lab, they also published, recently one paper published in Nature, the role of pericyte in the blood-brain barrier integrity, how pericyte loose or the marker affect the blood-brain barrier integrity. They showed the PDGFR beta, role of PDGFR beta in that.
But these studies they don't look at specific location of the capillaries. I'm assuming they're examining
The
function.
in my TBA model, this is focal injury model. So we induce injury or inflict injury in one area of the brain, mainly parietal lobe. So because of this focal injury, we cannot see the effect of injury at other part, like very close premises, close proximity of the injury area, we can see the good result. So that means sampling is very important in this model. Like if we collect samples from far away from the injury, we cannot see. So, but like unlike like in diffuse injury, TBI, we can see like the result in all of the brain. This is focal injury. This exactly mimics car accidents and falls. So here, for your questions, we induce injury in the parietal lobe because initially we did like prefrontal cortex, and we move to parietal lobe because we want to analyze some cognitive functions in the hippocampus because hippocampus is just below the parietal lobe.
I have one question. My impression that all these analyses were performed at the level of capillary bed. Do you also explore precapillary arterioles, what you have as muscle cells included in the neurovascular unit? Yes, I guess from the diameter of the vessel that you presented, you are looking at capillaries.
This is capillary. Yeah.
Have you seen any changes at the level of precapillary arterioles, bigger, that have the small muscle cell as part of the unit?
Not much like we did study because we mainly focus blood-brain barrier. Of course, the big capillaries or brain microvessels. We did this study in alcohol actually. In TBA we didn't do that. One paper in 2017 we published with a clear showing the picture of microvessels, capillaries, and small capillaries. But in TBA we didn't study. It's a good question. There's a role for study.
Any other question? Yes.
I wanted to ask, so I'm not sure how is the treatment administered to the rats, and how do you expect treatment would look like in
Keep treatment. You are asking like vector treatment, right? This is systemic treatment, like a subcutaneous injections, But we directly inject to the brain. Adeno-associated virus. CRISPR we directly injected to. If you are asking about peptide we injected subcutaneously, and we peptide joined with the TAT sequence. Because of TAT sequence, it crosses to the blood-brain barrier and it penetrates to the cells.
Any further question? No? So we thanks Dr. Muneer again.